So the idea of this is to kind of share with the community the importance of creating a patient-derived inducing pluripotent stem cells, or IPSC for short, bank. And this, the fact that we are organizing this kind of questions and answer session right now is because we want to make the most of the clinic that you're going to have, the foundation is going to have, so we can try to recruit as many patients as we can in order to collect the samples that we need from the patients and then generate this IPSC bank. So with the hope of just making this very short, to the point, and very understandable, I just have one big slide with a cartoon that is going to explain basically what are IPSC, what is a bank that will host this IPSC, and how will that help the community in general, not only the patient community, but also the scientific community and clinicians. So here, this is like a very general cartoon about generating iPSC cells. And the idea is that we can collect samples. And in this case, I'm just talking about a blood draw. This is the particular sample that we're going to collect from patients from a very diverse population. We all know and we're aware of the differences in symptoms and manifestation of CAT6A and CAT6B disorders amongst our children. So one of the questions that we want to tackle is, why do we have all this variety and this diversity of clinical manifestations? So for that reason, that's one of the reasons that it's very important to collect as many samples that we can from different patients, so we can have a good and accurate representation of all the community. The other reason why we want to collect samples from different patients is because even though this is not originally thought for achieving precision medicine, medicine that will specifically work for one patient, this is a step closer. So we will have the tools already for researchers to perform all the experiments needed in those cells that are going to be patient-specific. And those cells are going to honor all the differences and particularities that patient genome is going to have. So for those two main reasons, we want to make sure that we can collect as many patients as possible. And there are another scientific other reasons, for example, where the pathogenic variant is located, in which part of the protein is affecting. But all those are scientific reasons that, even though are going to have a positive impact in the population and in advancing research in CAT6A and CAT6B related disorders, are a bit tangential to the greater good. So iPSCs are induced pluripotent stem cells, and we all know, we are all a little bit familiarized with stem cells, which are embryonic cells that have the ability to generate every other single cell of the human body. And here we are talking of generating from a blood draw cells that are going to be similar to those. And the way this happened is we draw blood from patients. This could be through blood drawn. Also, skin biopsy is also another method that is used for generating these specific cell types. From that blood, we're going to isolate what we call peripheral blood mononuclear cells, or PBMCs, and those cells are already differentiated cells from that specific patient that have all the genomic background of that patient, and we have the ability of put those cells through a process that is called reprogramming. And with this process, what we say to this blood cell is, we want you to be as similar as possible as you were in an embryonic state. So with this process, we're giving these cells that are already differentiated, when we are taking them back into a state where they will have the ability to differentiate into several tissues, and hopefully into several cell types, and hopefully, depending on the experiments, in tissue organization. So when I'm talking about iPSC, I'm talking basically about cells that are very similar to stem cells, that will have the ability to generate different tissues, the different cell types and tissues. So that's basically an iPSC. And what's wonderful about this is that we can achieve this quite easily by just drawing blood from patients. Once we have this collection of stem cell-like cells, or iPSC, from many, many different patients, that means that we'll have different kind of pathogenic variants. We're going to have also different kind of genetic background making a good representation of the whole population. We can store those cells in what is called a bank. Very similar to the concept that we know about banks, is a place where we can safely store these cells. We're going to reprogram them, characterize them very well, make sure that these are in good conditions to share with the scientific community. There are a few controls that we need to run just to assure that these cells are properly reprogrammed. And once we have all those controls, those cells get stored in a bank. And this bank is hosted in the Center for Regenerative Medicine, which is now my home. And one of the wonderful things of this center is that this bank is maintained by a grant that allows the shareability and distribution of these resources without charging anybody for sharing them. We only charge a very small fee for maintenance of the bank, and then an MTA agreement signature, which is something needed to make sure that we are sharing the resources in the appropriate framework, scientific framework. So we are just offering a place where we are going to grab these blood cells, reprogram them, make sure that they have the ability to generate every other single cell type of the body, and then we're going to store them in a very precious way. So then we can maintain them and have this catalog of different cells coming from different patients. These cells can be shared. Here I'm saying worldwide because in theory that's the concept. Any other researcher, academic institution, or even industry who wants to use these cells, they can just request them, and we make them sign an MTA, and then just a minimum fee, and those cells are shipped. But the truth is that for being able to share them worldwide, we do have some problems with borders and custom depending on the country. So the restrictions some restrictions might apply depending on the regulation of each country. But theoretically and practically in the US, they can be shared without any kind of restriction. And then worldwide, in theory, they can be shared, but we just need to be aware and conscious about every country, or all the regulations that every country will have, different countries will have. And then, since these cells are going to be available for every researcher that wants to pursue research in Cat6A or Cat6B, we know that these cells have the ability to generate different cell types. These cells can be used to grow cardiomyocytes or neurons or lung cells or gut cells. They can be used with different protocols for inducing differentiation of specific cell types. And with a step forward, we can also generate what are known as organelles, which are 3D structures of those cells that resemble somehow the structures of the organs in developmental biology trend. For example, currently we know in my lab we are doing brain organoids from neurons, so it's just a neuronal tissue that undergoes a specific process that allows them to form 3D organs, and that organoids. And that's something very good because it's taking us a bit more closer to what we know as translational medicine, and we can ask questions that then can lead us to the possibility of discovering therapeutic approaches or even test hypotheses that we currently have in our field. So the idea is that having the possibility of testing different protocols and even drug screening with all the cells from different patients will give us not only one drug that might work for only one patient, but the possibility of drugs that will work for every single patient, that will work for all. And all of this is if every lab wants to do this as an individual effort, it's very time consuming and extremely expensive. And here we have the possibility, which I consider very unique, of doing this in a non-expensive way, because one of the goals of the Center for Regenerative Medicine is to share these resources. So what is this? What is the Center for Regenerative Medicine, and how do we know that there is a catalog of cells, and how can we validate or verify this? So there is a webpage for the center where you can actually go and check the catalog of cells that are currently being hosted in the bank. It's just crem.bu.edu. And here I'm just showing you a little bit of the webpage. One thing that is super important is this open source biology statement, which is by which we commit to share every single resources that is generated by our center. Everything we generate scientifically is a resource that is going to be shared, including these IPSC cells. Then you can go to the top, go to course and protocols, and there you will have access to the core facility, which is actually going to generate this IPSC for no cost for the patients. And you can just go to the clip where it says bank, and you will see this catalog where you can go to disease, and you will find a wide array of diseases that are already hosted here. So I invite you to go and to check here. Cat6a, Cat6b is not there yet in the catalog, but our goal is to include this. And then every researcher, family, patient can just go over here and they can see and check, and not only the availability of those cells, but also some clinical information and some genetic information that is going to go together with those cells. So one example of what's the potential of this research. And here I'm just sharing my research program, in which I use cells that in this case are, in the case I'm going to show you right now, are genetically engineered to be to have a pathogenic variant in KMT2B. So these are CRISPR-Cas9 modified cells, but we are also working towards developing iPSC cells from patients. So once we obtain these cells from either genetic engineering or from patients, we will generate these iPSC cells that are stem cell-like cells that allow us to generate every single tissue. In my case, in my lab, we differentiate this in neuronal tissue that produces this rosette structure. This is neuroepithelium that arrays itself in a concentric way. And then we grab each of these rosettes and generate brain organelles, or we do two-dimensional culture. And in these models, we test not only different genetic tools, but also drugs and drug screening. And then we perform a wide array of techniques that allow us to understand not only the response of those cells to specific drugs, but also what is the fingerprint, the molecular fingerprint that is leading to what we observe in the clinic as a phenotype, as the way that a patient is manifesting the disorder, in this case, Kabuki syndrome. So here, this is a cartoon, and now here I'm showing you the actual organoids. And you can see these are called minibrains sometimes, but they are not. You can see it's a very primitive structure. It's just a bit of neuroepithelial organized in a three-dimensional fashion that has the ability to experience the process of neurogenesis. So we have neuronal progenitors that then differentiate into mature neurons, and then we can study neuronal connections and synapses and test different hypotheses. But that's how it goes from day 1 to day 14. So here, once we have these cells and the possibility of having a stable colony of iPSC, I'm telling you that in just 14 days, you can have already material to study and to test hypotheses, in this case, in order to understand what are the causes for the phenotypes that we observe in Kabuki syndrome. And well, one of the things that we want to also know is, like, is this actually a system that is allowing us to reproduce what we observe in the clinic? And we can see the difference between the the samples, or in this case, the brain organelles that do not have a pathogenic variants, and compare that with the ones that do have a pathogenic variant. And for example, here at day 14, you can see that the disease scenario has these brain organelles are smaller and have a different morphology to the one that is a non-disease scenario. And of course, you can quantify that, and it's very straightforward to assist model system to continue asking questions in terms of the biology of this of Kabuki syndrome. But I'm not going to bore you with details about this. I just do want you to know that we have the ability to use these structures that just came from blood. Again, this is super important. From blood, we're generating these cells that have the ability to then generate any other, potentially any other cell of the human body. In this case, we're generating neurons. Here, this is a non-disease scenario. We have these progenitor, neural progenitors that will then differentiate into different neurons. And over here, we have axons of more mature neurons. And you can see how in the pathogenic variant scenario, we do not have the formation of axons amongst these neural progenitors. So you can see already a difference. And we can understand why this is happening molecularly and cellularly in these brain organs, and everything from just the willingness of patients to contribute to this research and helping us to help understand the community about what's the best, what are the best avenues to explore in order to test different treatments and understanding the clinical manifestations. And then we perform, probably many of you already heard about single-cell RNA sequencing. These are very important techniques that help us researchers to understand what are the differences in specific cell types. Because yes, I'm telling you neurons, but not every neuron is the same. Neurons have specific functions, and if we want to really understand what's the particular neuron or the group of neurons that are probably not having the ability to, I don't know, make synapses or connect or differentiate, or probably some neurons are inexistent in these organs with the pathogenic variant. So then we have the possibility to identify those at the transcriptome level, at the molecular level with single-cell RNA-seq. And here, for example, in this cyan or light blue, you see the disease scenario, the organism that has had a pathogenic variant of the protein that causes Kabuki syndrome. And here in this pinkish red color, we have the non-disease scenario. And each of these dots is one cell of a brain organoid. And you can already see that the non-disease scenario has cell populations that are non-existent when we have a Kabuki syndrome brain organoid. So, and we have the ability currently to understand what are these neurons that are missing, what's the consequence of this in the clinical manifestation of the patient. So as you can see, this is extremely powerful. And we can do exactly this from just one patient donating blood to just the ability to share this with the whole research community. And this is just one example. There are people that are working in different cell types in order to understand different clinical manifestations of the disorders. And with that, I will put again, I don't know if I have, I'm just going to put here super quickly my email in case that anybody watching the video might have any questions. Sorry, double R. So please feel free to contact me, and to I'm more than happy to talk to everybody and try to respond in the best of my ability any questions that anybody might have. But meanwhile, I don't know if any of you have now any questions.